you've heard a lot about how we generate mutants, how we analyze phenotypes, and so on. And we have a number of talks where we look at how we use the mice in various translational domains to understand how biological systems work, particularly disease systems, and how we can use the mouse as a model in translational research to find new therapies. And what I want to focus on today over the next 30 minutes or so is the use of mouse mutants to study this disease here, which I'm going to tell you a lot about in a minute, chronic otitis media. It's a disease of childhood. It's a severely debilitating disease, enormous healthcare costs involved, but we know very little about the underlying molecular mechanisms of it. And equally, we have very poor treatments. And I'm going to tell you a story about using two mouse mutants, principally Junbo and Jeff, which model this disease and have allowed us to identify the underlying pathways involved in chronic otitis media, and through that, identify novel therapies. So otitis media, put very simply, is an inflammation of the middle ear. The middle ear cavity is an important aspect of auditory transduction. Sound comes through the external ear and the external ear canal to the tympanic membrane, vibrates the tympanic membrane, and moves these ossicles in the middle ear to transmit pressure waves into the inner ear, where the process of mechanotransduction takes place, by which auditory sound signals are converted into neural impulses to the brain. But the middle ear plays a very important part. It's the transducer between the sound signals in the external environment and the inner ear. And one important aspect of the middle ear cavity, it must be, as air pressure in the middle ear cavity must be equalized with the outside to function properly. And it's equalized by the eustachian tube to the nasopharynx. And we all know this because when we go up in a plane, pressure doesn't get equalized in the middle ear. And you all know this and experience this feeling of sound being damped in the middle ear because pressure between the middle ear and the outside world across the tympanic membrane is not equal. That means the tympanic membrane doesn't work properly and auditory transduction is impaired. Oop. And of course, that can also be impaired when you get a bad cold. In the nasopharynx, you get fluid which migrates up the eustachian tube. It fills the middle ear cavity with fluid and pus. And that also leads to some sensorineural hearing loss. You've all experienced this effect. But of course, what happens in many children is that they not only they get this fluid-filled otitis media in the middle ear cavity, not just when they have a cold, but through other factors, including bacterial infection of the middle ear, but also this chronic disease, which I'm going to describe in a moment, called chronic otitis media. So there are really two major types of otitis media. There's what's called acute suppurative otitis media. 2/3 of children by the age of 5 will suffer from episodes of acute otitis media, which is due to bacterial infection ascending the eustachian tube into the middle ear. You get an infection in the middle ear, you get a chronic suppurative disease, you get an acute suppurative disease going on, which usually clears after about 8 or 9 days. It leads to a significant number of deaths around the globe, mainly in the developing world. And sometimes this acute suppurative infection can become chronic, and this affects a huge number of people worldwide and causes the death of around about a further 28,000 people a year globally. But I'm not really going to talk about the acute disease today that arises from infection of the middle ear. I'm going to talk about another very serious type of otitis media, which is otitis media with effusion. And this often follows an acute attack, a bacterial infection of the middle ear. 95% of children by the age of 5 will have some period where they have an otitis media with effusion. It's a blocked middle ear. There's not an ongoing infection. The middle ear is just full of pus and fluid, which doesn't resolve immediately. This is sometimes known colloquially as glue ear, and of course it significantly impairs hearing in these children. It's in fact the most common cause of hearing loss in the world, And the only treatment for it at the moment is to insert grommets, a surgical procedure that requires a general anesthetic into the children to treat this chronic disease. And inserting grommets is, in the US, for example, there's 2.2 million general anesthesia episodes every year in the US at a cost of some $4 billion. So it's enormous healthcare costs. Of course, there's a big cost to the children because children who develop chronic otitis media with effusion, although 95% of children by the age of 5 will have some otitis media with effusion, in most children that resolves. But many children go on to develop a chronic form of the disease, and it's these children who require the insertion of grommets, ostensibly to relieve pressure in the ear, to drain fluid, but nobody really knows how grommets work. It's basically a sort of middle-aged therapy that people found has some relief, but it's relatively ineffective. Grommets often drop out, and you have to have another surgical intervention to put them in again. Of course, children who have chronic otitis media with effusion, irrespective of whether they have surgery or not, it often resolves. And of course, it always resolves by late childhood and teens. So this isn't a disease that you really ever see in teenagers or, for that matter, adults. So we have a very common disease with high healthcare costs and also enormous costs on the children. Because children with chronic otitis media that lasts over many months, perhaps years, this glue ear, suffer from language impairment, social isolation, and many other factors that affect their development. So we have a very common disease, high healthcare costs, and poor treatment outcomes. So we need to do something about this, partly because we don't understand why some of these kids develop this chronic otitis media. Why does it happen? What are the mechanisms? So if you look at otitis media generally, as I've described, there's a whole number of different types of otitis media. On one side of this graph, going in this axis, we have inflammation. Here we have the type of effusion, from a very suppurative, granulocytic effusion, a mucus effusion, a subclinical effusion. And on one side, if you like, of this set of otitis media conditions, we have those acute otitis media, which I've already described, which reflect an infection episode in the middle ear. But over on this side of the otitis-prone spectrum, we have inflammatory conditions that arise presumably due to perturbations in the ability to control the inflammatory response in the middle ear. And of course, in one of these major conditions, chronic otitis media with effusion, COME, that's one of the major things that we're interested in. But it's over this side, understanding what leads some children to develop an inappropriate chronic inflammatory response that is not appropriately resolved, goes on, gives you glue ear, impaired language development, social isolation, and ultimately impaired development for those children. But the major point to take home from all of this introduction is that we know from studies of human populations, we don't know the genes involved, but we know that genetic factors are known to play a major role in the recurrent and chronic forms of otitis media with effusion in the human population. But no genes, I stress no genes, have been identified. So if we can find the genes, we can find the mechanisms, we can develop new therapies. So this is where these mouse mutants have come in. I'll go through the 2 mutants that I'm going to focus on very quickly. We have more mouse mutants, and I'll tell you about them later. But the mouse mutant Jeff is the first one we discovered over 10 years ago. It's a single gene model for chronic otitis media. It's identified from an ENU program. It shows hearing loss, and it has a hearing loss due to conductive deafness due to the development of a chronic serous otitis media. Now, this is a cross-section across the middle ear. You can see the tympanic membrane here. Here's a normal wild-type mouse with a clear middle ear, no effusion whatsoever. Here's the Jeff mouse. You can see that the middle ear is full of pus and fluid, and that is what's leading to the hearing loss in this particular mouse. So when we found this mouse, we thought this is a way of deciphering what might be the genes involved in chronic otitis media. So the gene that, in fact, underlies this mutation is an FBOX11 gene. I'll tell you a bit about FBOX genes in a moment. In fact, we have two alleles. This is the mutation here. It's a non-conservative glutamine to leucine mutation. In the middle of the gene, we have another allele over here. And we published this in 2006. So what do FBOX genes do? Well, in fact, FBOX genes are an important part of protein turnover and protein degradation because they act as specificity factors for a group of E3 ubiquitin ligase, the SCF class of ubiquitin ligase. And what they do is that they bring particular substrates to this E3 ligase complex so that they can become ubiquitinated, particular protein substrates, which get ubiquitinated and then degraded. So these FBOX genes, there are many of them. There's 74 in total in the mouse genome. They're responsible for bringing specific proteins to the protein degradation pathway and turning them over. So the other gene that I'm going to tell you about today, the other mutant that I'm going to tell you about today is very similar. It's called Junbo. It was also identified from the ENU mutagenesis program. And again, it's deaf because it's got a conductive deafness due to a chronic suppurative otitis media. And again, if you look at the wild type middle ear here, you can see a clear cavity. And here's the Junbo middle ear cavity. You can see that it's full of fluid and pus. And that's what's leading to the hearing loss in these mice. So a second mutant involved in chronic otitis media. So Junbo is a mutation in a transcription factor known as EVI1. And in fact, this transcription factor has got a series of zinc finger domains, ZF1 and ZF2. And the mutation, in fact, asparagine to isoleucine change in a highly conserved residue in this second set of zinc finger domains in this protein. So what do these proteins do to cut? After a lot of investigation, we know a lot more about what they do. EVI1, I'll start with that, the Junbo mutation, is in fact, this transcription factor is a repressor of SMAD3, and SMAD3 is a critical pathway member in Tgif-beta signaling. And interestingly, FBOX11 also signals through this path, through the Tgif-beta signaling pathway as well, because FBOX11 is known to be a repressor of P53, which interacts with another signaling molecule in the Tgif-beta pathway, SMAD2. So both of the interesting thing that immediately we found out about both of these proteins is that they're both impacting upon Tgif-beta signaling. And this immediately gave us a sense of what these two proteins might be doing in terms of affecting the inflammatory processes in the middle ear. And the reason is that there's very strong crosstalk between the Tgif-beta signaling pathway and the hypoxia signaling pathway. Indeed, SMAD3 itself is a repressor of an enzyme called prolyl hydroxylase D2, which in fact is a repressor of HIF1 alpha. HIF1 alpha is the major transcriptional factor that sets off the response of tissues to the lack of oxygen, the hypoxia signaling pathway, and I'll tell you a bit more about this in a moment. But this is just to give you an overall picture at the start that in fact both of these molecules involved with chronic otitis media seem to signal through Tgif-beta signaling pathway, and our hypothesis was that they would impact upon hypoxia signaling. So there's interplay between the Tgif-beta and hypoxia signaling pathways, and we wondered whether dysregulation of Tgif-beta signaling in Junbo and Jeff impairs the response of the middle ear to hypoxic conditions. Because if you think about it, the middle ear, it's a poorly ventilated space. It's full of pus and glue. So the tissues there are going to lack oxygen. They're going to lack the ability to be ventilated through the eustachian tube. Hypoxia will result. And that impairment of the response to the hypoxic conditions might be critical in terms of making these mice more susceptible to the development of chronic otitis media. So we went and looked at the middle ears of our mutant mice, both Junbo and Jeff, And we stained them with this label, which actually identifies hypoxic tissues or hypoxic cells. So this PIMO label, you can see, is lighting up this is the epithelial lining of the middle ear. And you can see in the center of the middle ear in jumbo mice, you can see the effusion, the granulocytic effusion. You can see macrophages here. You can see that they're all staining with PIMO, demonstrating that the aeration of the tissues in the middle ear of jumbo mice is impaired, that they are indeed hypoxic. You can see the wild-type control here, a very thin, uninflamed epithelial lining with no staining with PIMO at all. If we compare the staining of jumbo and wild-type mice, you can see that jumbo clearly shows, as it develops, extensive hypoxia developing. So this led to a model of how hypoxia and dysregulation of hypoxia in the middle ear leads to this chronic effusion and inflammation in the jumbo mice, arising from the interactions between HIF and TGF-beta pathways. So here's our model here. Jumbo is a mutation in EVI1, which is a repressor, as I've already said, of SMAD3. So if we remove jumbo, SMAD3 is increased. SMAD3 represses PHD2. If SMAD3 increases, PHD2 is further repressed. And PHD is responsible for turning over and reducing HIF1 alpha in response to hypoxic conditions. So HIF1 alpha goes up as well. HIF1 alpha and SMAD3 are both raised. And both of these proteins raised in the cells of the middle ear are going to prime the transcription of a factor called VEGF, vascular endothelial growth factor. And VEGF is an important signaling molecule for angiogenesis, the formation of blood vessels, an inflammatory response, and vascular leak. So this is our hypothesis, that through the mutation of EVI1, we lead to an inflamed middle ear epithelial lining, more blood vessels, an inflamed mucoperiosteum around the middle ear, vascular leak, fluid leaking into the middle ear cavity, with the result that we get a chronic otitis media. So we went to investigate this hypothesis, which indeed we found to be the case. So if we look at the genes that are being expressed in the middle ear, we find that many of the genes, including HIF and its target genes, as well as VEGF, that key molecule to set off this process of inflammation and vascular leak into the middle ear, they're all elevated in the middle ear of jumbo mice, and they're elevated in the middle ear of Jeff mice as well. So that's the first part of the hypothesis proven. And in fact, if we look at levels of VEGF protein, they're elevated in the middle ear fluids of jumbo and Jeff mice. You can see here that if we look at jumbo ear fluid, you can see the enormous levels of VEGF protein that we see in the middle ear. The same is true of Jeff. And this protein is really responsible in all tissues for angiogenesis, vascular permeability, and vascular leak. So our hypothesis is that those raised levels of VEGF are pushing fluid out into the middle ear cavity, creating the effusion, and at the same time, the angiogenesis is leading to the extra blood vessels and lymphatics in the epithelial lining, which is part of the inflammatory process. So if VEGF's upregulated, VEGF signals through a Vegf receptor. Here's VEGF here. It binds to Vegf receptor, and it sets off a whole signaling cascade, which leads to the effects that I've just described, including angiogenesis, vascular permeability, and so on. So if we look at all of the genes in the VEGF pathway, are they upregulated in Jeff and jumbo mice? And indeed, we looked at the transcripts of all these genes in the middle ears of Jeff and jumbo mice. And I'm not going to go through the details here, but we used what's known as an RTQ PCR signaling array. We found a whole slew of genes that are significantly upregulated, more than twofold higher than you would normally expect. These are all the genes here that are upregulated. Many of them are in the VEGF signaling pathway. So this is our model, basically, of what's happening. Just to return to it, here's the normal middle ear cavity here. In normoxic conditions, you have the bone, the overlying, what's known as the mucoperiosteum, the epithelial lining. And what we're assuming is happening in Jeff and Junbo is that you get an effusion occurring in the middle ear, maybe some vascular leak, which is set off by maybe an environmental irritant or whatever. That leads to VEGF being produced, which signals to the mucoperiosteal lining for angiogenesis, lymphangiogenesis, and vascular leak. More fluid, more inflammatory cells. In these mice, we get a chronic runaway inflammatory response to the hypoxia that's developing in the middle ear that's not properly controlled. So the question was, how do we prove this mechanism? And the second question is, if this is the mechanism, it appeared to us that that implies that we could potentially, it provides new target pathways for therapeutic intervention. Because the exciting thing about finding that VEGF is a clear player in the chronic otitis media for these mutants is that for a variety of reasons for cancer therapies, there's been a whole slew of drugs and compounds that have been designed to inhibit Vegf receptors and to control the response of cells and tissues to VEGF. So our thinking was, if this is the mechanism by which we're leading to chronic otitis media, that perturbations of TGF-beta signaling manifest themselves through the hypoxic response of HIF-1 alpha and VEGF signaling, then we could intervene in this pathway by using compounds that inhibit the VEGF response, that target VEGF receptors and damp down the response that you get from increased levels of VEGF. So in fact, there's a whole number of drugs that are off patent now, actually, that you can use. They're used in a variety of cancer therapies. They all impact on VEGF receptor to lower angiogenesis, to lower vascular permeability, to lower vascular leak. And we looked at 3 compounds, vitalanib, sorafenib, and sunitinib. They all, as I said, act on VEGF receptors, and they're classically anti-cancer drugs. Their action is anti-angiogenic, anti-lymphangiogenic. We also used another compound, which in fact would impact upon this pathway, and this is a compound called 17-DMAG, which to target VEGF receptor signaling as well. This inhibits a chaperone called HSP90. HSP90 chaperone is a chaperone for HIF-1 alpha, which is involved in hypoxia. And normally it destabilizes HIF. So we wanted to use that particular compound as well to see whether we could intervene in the pathway there. So the experiment that we set up was to take our modeled jumbo mice and to take our VEGF receptors, and at 28 days, we systemically administer to the jumbo or wild-type mice these VEGF receptor drugs to see what would be the response to the chronic otitis media that's developing in these mice. So we administer the drug to the mice, and then after a month's administration, we actually look at the hearing in these mice. We measure something called the delta ABR. And all that means really is that we look and see how are the hearing thresholds of these mice developing. In the jumbo mice, untreated, they will get worse and worse. Gradually, the middle ear fills with pus and fluid to the point where they have a very significant hearing loss and become deaf. We're simply measuring, can we stop this deterioration in ABR thresholds of hearing? We also looked at the histopathology of the middle ear to see whether we see the kind of inflammatory changes that we see with Jeff. Can we abrogate, can we damp down those histopathological changes that result clearly from increased VEGF signaling? So the answer is yes. I'll just take you through the results of this experiment because this shows the 4 compounds that we used, vitalanib at 2 different concentrations. And for each of those compounds, I've displayed what happens if you give those compounds to wild-type mice, if you give a sham treatment, and if you give the drug. And the axis here is what's called the delta ABR. It's the change over that month in which we treat the mice in terms of deterioration of hearing. First of all, let's look at jumbo mice treated with sham, which is indicated by green. You can see that their ABRs deteriorate, all of them. But if we treat these jumbo mice with drugs, these are the blue bars that you need to look at, you can see that there's a significant improvement in all cases in the delta ABRs. And in some cases, such as with sunitinib, you can see that there is no deterioration whatsoever in the ABRs over that month. We've effectively stopped the disease in its tracks. The chronic otitis media is not developing. So this was a very exciting result for us, not only because it suggested a new treatment, but it suggested the mechanism that we had hypothesized from our studies, the mechanism of the chronic otitis media, which is an inappropriate response to hypoxia through VEGF signaling, is the right hypothesis. So VEGF receptors moderate hearing loss, and of course that identifies a potential new therapy. Just to emphasize again, I just put this bar down by a 0 delta ABR. This would be no change. You can see that the wild-type mice are showing no change in their hearing thresholds. And in many cases for these drugs, we're getting the mice back down to a 0 delta ABR, in contrast to the sham-treated mutants, where the hearing continues to deteriorate. We can also look at the mucosal changes in inflammation, not just the actual hearing thresholds in these mice. So here's a wild-type mouse. You can see the empty middle ear cavity, no effusion, a nice thin epithelial lining. Here's the jumbo mice at 56 days. Look at this enormous effusion in the middle ear. Look at this incredibly inflamed middle ear epithelial lining with fibroblast proliferation, the polyps extending into the middle ear cavity, accompanied by significant angiogenesis and lymphangiogenesis as well. So we can measure how the VEGF inhibitors moderate mucosal proliferation. I'm only showing the results for vitalinib here. For both blood vessels and lymphatic vessels, again, you have the drug administered to wild-type mice. There's really no change in the blood vessels and lymphatics, of course, in wild-type mice. And then the drug and sham treatments in jumbo mice in terms of the number of blood vessels and lymphatics. And you can see that there's, in the drug-treated jumbo mice, there's a substantial improvement in the number of blood vessels and lymphatics that we see in the middle ear epithelial lining, showing that, again, we're moderating mucosal proliferation. And also you can see this from the perspective of drug and sham-treated mice in the amount of fluid in the middle ear as well. No fluid is blue, fluid is red, and you can see that in the drug-treated mice, we have a significant improvement in the levels of effusion in the middle ear. So overall, VEGF receptor inhibitors seem to be playing a role in damping down otitis media. So we have a new therapeutic route for chronic otitis media through the targeting of VEGF receptors, moderating hearing loss, reducing fluid, and reducing the inflammation in the middle ear epithelial lining in terms of the formation of new blood vessels and lymphatics. So this was very exciting for us because, of course, it immediately suggested a new way to treat chronic otitis media, which we're now working on. We've also been collaborating in this respect with AstraZeneca and looking at other compounds that might target the VEGF receptor pathway. We need to establish the efficacy of new drugs that we're getting from AstraZeneca. And importantly, of course, because most of these drugs are quite severe compounds with significant side effects, if delivered systemically to children, this would be the last thing we want to do. We need to design ways of getting these drugs into the middle ear ototopically, directly through the tympanic membrane. And indeed, we're doing a lot of work on that at the moment. We formed a new company to take this forward because, of course, while we wouldn't want to give these drugs systemically to young children, the middle ear cavity is relatively amenable to drug delivery through the tympanic membrane by various routes that we've designed and thought about. Watch this space, as it were. So clearly we're thinking about how to deliver these drugs ototopically directly to the tympanic membrane, thus avoiding the use of grommets, which, as I said, don't work very effectively, but also avoiding the use of surgery. We would deliver these drugs across the tympanic membrane, probably in the primary care scenario. So children would go to their GP, they'd get some patch or something put on the tympanic membrane, which would deliver drugs across the tympanic membrane, damp down the effusion and the hearing loss. And one of the things we're very focused on with AstraZeneca is also finding drugs which that act on this pathway, that don't just stop the chronic otitis media from developing, but actually reverse it as well. So one of the questions that I'm sure you're asking is, well, what happens in children? Do their effusions, does the molecular mechanism in humans look the same? And in fact, we've been able to study this because we can get samples of human middle ear effusions from children who are undergoing grommet surgery. And we've looked at the effusions to see whether the same mechanisms appear to be operating in humans. And the answer is yes. We see the same upregulation for key genes, such as VEGF and HIF1 alpha, which are clearly found in our mouse models. So the findings in humans appear to mirror those from mouse models, so that we would expect the treatments that we've devised in the mouse to work in humans as well. So future developments are to further elaborate the mechanisms of chronic otitis media. We need to investigate additional inhibitors and small molecules, as I've already indicated. We have several new mouse mutants available, and I'll say a little bit about that in a second. But we also want to further investigate the underlying molecular basis of human chronic otitis media. But this is the key thing. We need to develop delivery systems for topical administration now to take this forward into clinical trials and ultimately to the clinic. I want to stay on time, so I was going to say, but I'll just skip through them, that, of course, we have other mouse models now for chronic otitis media. This is one gene, I won't tell you much about it, transforming growth inhibitory factor 1, which works in the TGF-beta signaling pathway. Again, this brings us always back to TGF-beta signaling. And these mice show the same upregulation of VEGF alpha and other cytokines in the middle ear effusions. We have another mutant recently described called Edison, which carries a mutation in this rather unknown gene called nisharin, but also appears to signal through TGF-beta signaling pathway as well as other pathways. But to cut a long story short, we now have 4 mutants, Junbo, Jeff, Edison, and TGIF, all of which provide us with a significant degree of confidence that the TGF-beta pathway, as well as some other pathways, plays an important role in the process of controlling hypoxia in the middle ear, perturbing it, resulting in significant dysregulation of hypoxia, upregulation of VEGF signaling, and all the chronic inflammatory manifestations that we see in chronic otitis media. So it's through the use of these mutants that we've really been able to move forward in terms of studying the molecular mechanisms of chronic otitis media and delivering new treatments. So the last point before I stop is, do mutations in any of these genes occur in the human population that also lead to chronic otitis media? And the answer, at least for some of these genes, is yes. So we've been carrying out population-based studies to see whether genetic variants in any of these genes seem to be associated with otitis media in the human population. And the answer is that for 2 of the genes, FBOX11 and TGIF, the answer appears to be yes. FBOX11 has come up in a number of association studies worldwide and seems to be associated with the disease in both US, Australian, and UK populations. And we have a big major human genetics initiative going on in the UK, the Genetics of Otitis Media Study, where we've collected over 1,500 families for chronic otitis media with effusion and analyzed them for all of these potential otitis media genes. And you can see that the 4 top hits are in these 2 mouse models, TGF1 and FBOX11. So these 2 genes are certainly associated with otitis media in the human population and give us further confidence that the molecular mechanisms in the human population are the same as what we're seeing in our mouse models. So just to leave you with those developments again, we're very excited by this, and I think for us, it's a very compelling example of how going from a mouse model for a disease where we knew nothing about the molecular mechanisms gives us molecular insights, leads to potential new therapies, and then back to humans again to confirm that those mechanisms are actually occurring in humans. So we have a virtuous cycle, if you like, of interplay between mouse and human genetics in terms of understanding the molecular mechanisms of disease and the development of new therapies. So it only remains for me to thank all of the people at Harwell who were involved in this work, and just to mention 5 people, Hilda Tatiosian, Mike Crompton, Michael Cheeseman, Mood Boot, and Haley Tara, who did the lion's share of the work that I indicated today. And thank you for your attention. Thank you.
Thank you.